Abraham, does everyone, if somebody drinks in the 3rd week of pregnancy, a woman, is she going to have a defective child?
Studies have said that it's very difficult to, at the moment, to guess that, to diagnose it. People have tried to look at the women's similar size, similar weight, similar even ethnic backgrounds. It's very difficult to know. We know that some of the, for actually most of the genes, there isn't one copy in the human population. There's different variants. And it doesn't mean that one will cause a disease. But in this case, we're probably talking about a predisposition. So if we have an enzyme that it's, let's call it a bit weak, and then it sees alcohol at the same time, then the initial weakness becomes a serious problem. Because alcohol now, it's reducing the amounts of retinoic acid, and the enzyme cannot cope with it. The protein cannot cope with it.
So some people may not have any problems.
Some people may not have any
and
problems.
Some people have serious problems and some in between,
yes.
depending on this predisposition. Do they know the percentages of
Actually,
distribution?
there's very few. We know what the different variants are in each population. The numbers are coming with all the genome projects. But we don't know how they relate to fetal alcohol. I mean, it's very difficult. You can do what's called epidemiological studies, which means statistics on the human population, and see which people seem to have a higher incidence of fetal alcohol. But it's never been tested directly in a test tube to see which variant is a good variant and which variant is a weak or bad variant.
But in working out the mechanics of this, there's a better chance of figuring that out, I assume, because you could then do, once everybody goes in, has a $100 test on their genome, and you get it all mapped out, and you can say, wow, you're got a really high risk, or you have a low risk. That's a possibility.
That's one of the ideas in the future. I mean, at the moment, we're getting to the point that for about 1,000, a little bit more, you can already know your genome. Not 100, 1,000.
Yeah, but the price is coming down.
But you still have to collect the knowledge about which one would be a good gene and which one would be a weak or a sensitive gene. Let's call it that way.
But all these kind of breakthroughs that you made, for example, on the substitution that alcohol takes a critical protein away from the development of vitamin A, that is a general thesis about competition for chemicals, etc., to make sure the balance is kept right at each stage, so you can almost pattern it. And then I could imagine that then you pattern it with variations relative to gene structures. I would think, I'm not sure.
I mean, all our life and embryonic development, for sure, it's based on this really fine-tuned regulation. It is quite amazing to what level it can go. Goes from the basic function of the gene to how stable is the protein. Proteins are destroyed on cue by some regulatory mechanism in the cell. So this is all regulated inside our cells. It's quite amazing. We're just at recent years, we're scratching really the surface on understanding how, from the moment the gene is told make a little bit of protein to the moment you say no more, and how everything is regulated, and we're still in the diapers and understanding this.
Yeah,
processes.
but it's exciting because it's like being on the forefront of a whole new age of science. I mean, you know, this molecular biological science. I remember when I was in university, one of my colleagues went into just transference. He was working on transference of protein across the cell membrane, and that was totally original at that time. And now you take it all for granted.
No. Now we want to know for how long that protein stays in the membrane, and how, what's controlling it. Yes, go across. No, go across. Let's destroy it. We need more.
I see, the whole regulatory system.
One side of the cell is different from the other side of the cell, even though it's round. Because one side looks at one type of cells, and the other side might be looking at other types of cells. So communication is very important.
I see.
So all this is regulated. It's
So
quite amazing that we're actually alive.
but it says to young kids and young students,
Yeah.
if you're thinking of a field that's exciting, this is really an exciting field.
It's very exciting. There's a lot of open questions.
And at Hebrew University in this center, is this really a forefront of your research in this area?
And the center,
Because you belong to the Lautenberg Center, is that it?
No, no.
No?
No.
Oh, yeah, it's another
I
one.
belong to the Institute for Medical Research, and the department is developmental biology and cancer research. But I have my lab. Like I said, this PhD student wanted to change her subject. At the moment, basically, all the lab now works on this question, different aspects of fetal alcohol. We have some collaboration with there's a doctor that works on the epidemiology of alcohol effects. Let's not forget that apart from fetal alcohol, alcohol is a very important chemical that affects our lives manifold. We're talking about alcoholism and all the problems it brings. There is also alcohol has an effect on increasing the risk for certain types of cancer. And I'm not talking the digestive tract, which gets exposed to alcohol every time we drink. There's other like prostate and breast cancer are also, alcohol works as a risk factor.
I see, so it brings all kinds of implications there.
Our basic research on understanding fetal alcohol might have implications for, let's say, cancer risk, because it might be the same mechanism. It might also involve retinoic acid. We know that retinoic acid, this vitamin A derivative, is in some cases prevents the cells from becoming cancer cells. And when it doesn't work, then there's a higher chance of getting a tumor.
Are you short of resources to do this kind of work, or is that pretty?
All our work is funded by competitive grants. So we're all the time writing grants and trying to get funded for our work.
And I know one of the complaints I always had is we spend so much time writing the grant proposals, we don't get enough chance to do the work.
The same hasn't changed. It hasn't changed.
All over the world
we
the
have
same.
to always
And who do you have Canadian collaborators?
We're starting a collaboration. We started a project where we're trying to look at what we call genetic predisposition, to understand the variants of the genes and which one might be a risky gene to have. And one of the world experts actually sits in the Hospital for Sick Children in Toronto.
Oh, really? What's his name?
Gidon Koren. He's a former Israeli.
I see.
He's a pediatrician. His specialty is toxicology during pregnancy. And we've been in contact with him he has access to children with fetal alcohol. So we're trying to test if our idea to identify risky genes is right. And then he will provide us the DNA samples to test it in actual patients and
see. That's
if
very
our
interesting.
idea is right or wrong.
And he works with children on fetal alcohol.
He's a pediatrician. That's his specialty.
Oh,
he works, and he's one of the important figures in the fetal alcohol field in the world.
Okay, I want to end with a lighter tone than alcohol syndrome, which is you're a photographer in the zoo, and you take care of animals once a week. You volunteer?
Yes, I've always been interested in animals, and
not just the cells.
not just the cells, and not just for research. And a few years ago, perhaps my wife wanted me out of the house for a few hours. But anyway, she showed me an advert. The zoo was looking for volunteers to actually do caretaking. And I've been working there for years. I actually did a zookeeper's course. So I'm actually also a zookeeper. And once a week, for 4 or 5 hours, I work in the zoo.
And that gives you a chance to take your photographs of
always
animals.
my camera on the shoulder. And if I see an opportunity comes up, start taking pictures.
Well, with that note, we'll end, and thank you very much, not just for the interview, but your great research that you're doing.
Thank you.